Good morning everyone. Today we are going to study about the synthesis and secretion of thyroid hormones. Before moving on to the topic, we have to understand certain basic points regarding the thyroid gland. So the thyroid gland is situated in the neck, below the larynx and in front of the trachea. So you can see the thyroid gland. Here you can see the trachea and larynx. So it is present in the neck, below the larynx and anterior to the trachea. It has two lobes, that is a right lobe and a left lobe, and both the lobes are joined by a structure known as isthmus. Now when we take a cross section of a thyroid gland, we understand that it is made up of a lot of acini or follicle. So here in this diagram you can see 3 acini or 3 follicle. So each acini, if you look, it is lined by cuboidal epithelial cells. So these are cuboidal epithelial cells lining each acini or follicles. They are known as acinar cells or follicular cells. You can see the lumen here. This lumen of acini is filled with a fluid called colloid. Colloid contains a glycoprotein known as thyroglobulin. Now what is the importance of thyroglobulin? We are going to study the steps in the synthesis of thyroid hormones. So we should understand that thyroid hormones are synthesized within this glycoprotein molecule called thyroglobulin. And at the end, we will cleave this thyroid hormones from the thyroglobulin and it is secreted into the blood. Next to the follicle, you can see a group of cells known as parafollicular cells or parafollicular C cells, and they secrete a hormone called calcitonin. So calcitonin is a calcium regulating hormone or calcium lowering hormone. So whenever the plasma calcium level is high, calcitonin will be released from the parafollicular C cells of thyroid gland and it will lower the calcium to the normal level. Now the hormones secreted by thyroid gland are thyroxine or T4, triiodothyronine or T3, and T3 is more active than T4. Then reverse T3, which is inert, then calcitonin, which is a calcium lowering hormone that helps in regulation of plasma calcium. Moving on to synthesis and secretion of thyroid hormones. So iodine is the essential raw material that is required for thyroid hormone synthesis, and the body gets, or the thyroid gland gets, this iodine from the diet. So when you consume a food containing iodine or iodide, so it will be broken down and it will be digested and absorbed. So the iodide enters the blood circulation. Then the thyroid gland or thyroid follicular cells will pick up this iodide from the blood circulation into the cell and will use this iodide or iodine to synthesize thyroid hormones. And it is taken, or iodine is taken from the blood circulation into the thyroid follicular cells as iodide. Iodide is a negatively charged ion. Then minimum daily iodine intake for normal thyroid function comes around 150 microgram. Next, steps of thyroid hormone synthesis. So you have to understand there are 6 steps in thyroid hormone synthesis, starting from iodide trapping. So iodide trapping means iodide is taken from the blood into the thyroid follicular cell. Then conversion of iodide to iodine, which is also known as oxidation of iodide. That is, the iodide is converted to iodine. Then organification of thyroglobulin, then coupling, proteolysis of thyroglobulin, secretion of thyroid hormones. So proteolysis of thyroglobulin means thyroglobulin is a glycoprotein. So proteases will be there which will cause digestion of this thyroglobulin so that we can cleave the thyroid hormones that is present within the thyroglobulin. Then later these thyroid hormones are secreted into the blood from the thyroid follicular cells. You can see a diagram showing the thyroid follicular cell. So this is a single thyroid follicular cell. Now you can see the lumen here. Lumen will be having the colloid. Then the membrane facing the lumen is known as apical membrane of the thyroid follicular cell, and the membrane facing the interstitial space that contain the capillaries, and that membrane is known as basolateral membrane. So you can see the basolateral membrane is having sodium potassium ATPase pump and sodium iodide symporter. So we'll move on to the first step in the thyroid hormone synthesis, that is iodide trapping, that is transport of iodide from the capillary or from the blood circulation into the thyroid cell, which is done by iodide pump or sodium iodide symporter. So symporter means what? Both sodium and iodide will be taken towards the same direction together. So that's why it is known as sodium iodide symporter. Now you can see the sodium potassium ATPase pump here. So what is the role of sodium potassium ATPase pump? See, so the sodium iodide symporter is taking sodium from a higher concentration to a lower concentration, that is along the concentration gradient. And this concentration gradient of sodium is actually maintained by sodium potassium ATPase pump, because it will take 3 sodium from the cell into the interstitial space and 2 potassium from the interstitial space into the cell. And this transport of 3 sodium from the cell into the interstitial space is actually against the concentration gradient, because sodium will be more outside the cell. So that is against the concentration gradient. So they require energy, and that is by hydrolyzing ATP. So this is an example of primary active transport. Now because of this pump, we are having more sodium in the interstitial space. So it is easy for the sodium iodide symporter to take sodium from the interstitial space to the cell along the concentration gradient. But the concentration of iodide is more in the thyroid follicular cell when compared to the capillary or when compared to the interstitial space. So the transport of iodide is against the concentration gradient, so it requires energy. So why the concentration of iodide is more inside the thyroid follicular cell? See, after the synthesis of thyroid hormones within the thyroglobulin, there will be hydrolysis of the thyroglobulin, releasing these thyroid hormones outside the thyroglobulin in the cell, and later it will be secreted into the blood. Then this thyroglobulin also contain monoiodotyrosine and diiodotyrosine that is formed during the synthesis of thyroid hormone. And this monoiodotyrosine and diiodotyrosine will also be released during the hydrolysis of thyroglobulin. Now an enzyme known as iodotyrosine deiodinase will act on this monoiodotyrosine and diiodotyrosine. That will result in the formation of iodide and tyrosine, which will be again recycled for the synthesis of new thyroid hormones. That's how the concentration of iodide is more within the cell. So the sodium iodide transport carries iodide against the concentration gradient. And when sodium diffuses from higher concentration to a lower concentration, then the energy released is used by sodium iodide symporter to drag iodide from the lower concentration to higher concentration. So that is how iodide is carried by sodium iodide symporter from the interstitial space to the cell. So this is an example of secondary active transport. The next step is oxidation of iodide. So iodide is transported from the cell into the lumen by a chloride iodide ion counter transporter known as pendrin. So counter transporter means both the ions will be taken in opposite direction. So that's why it is known as counter transporter. If it is in the same direction, then we call as symport. Then iodide is oxidized to iodine by an enzyme called peroxidase and along with hydrogen peroxide. So here you can see now our iodide is present within the cell. With the help of sodium iodide symporter, we have taken iodide from the interstitial space into the cell. Now it is present within the cell. Now from the cell it is carried to the lumen with the help of chloride iodide counter transporter. So you can see both are taken in opposite direction. Iodide is taken from the cell into the lumen and chloride is taken from the lumen into the cell. So that counter transporter is known as pendrin. And also there will be oxidation of iodide to iodine. Okay, the iodide is converted to iodine by the enzyme peroxidase and hydrogen peroxide. The next step is organification of thyroglobulin. So that is binding of iodine with the thyroglobulin. So we know that thyroglobulin is a glycoprotein, so it will be made up of a lot of amino acids. So it's contain many amino acids including the tyrosine, cysteine, etc. Anyways, it contains a significant number of tyrosine residues. So organification of thyroglobulin means binding of iodine to the thyroglobulin. To be more specific, it is the binding of iodine to the tyrosine residues present in the thyroglobulin. And the enzyme mediating this step is thyroid peroxidase. Here you can see the diagram. Iodine is attached to thyroglobulin. So this step is known as organification of thyroglobulin, that is attachment of iodine to tyrosine residues present within the thyroglobulin by the enzyme thyroid peroxidase. And after binding we get a product. The first product formed is monoiodotyrosine, that is a single iodine is attached to tyrosine, that is monoiodotyrosine or MIT. Then if it is again iodinated, there will be 2 iodine, so that is diiodotyrosine. So 2 iodines attached to tyrosine residue, that is DIT. The next step is coupling reaction. So we know that monoiodotyrosine and diiodotyrosine is formed within the thyroglobulin. Now there will be oxidative condensation of 2 molecules of diiodotyrosine that results in the formation of T4. When there is condensation of 1 monoiodotyrosine with diiodotyrosine, that results in the formation of T3, that is triiodothyronine. Then when there is condensation of a diiodotyrosine with a monoiodotyrosine, you will get reverse T3, which is inert. So here you can see this is organification of thyroglobulin. Then within the thyroglobulin there will be coupling of MIT and DIT, resulting in the formation of T3, T4 and reverse T3. So this thyroglobulin will be containing T3, T4 and reverse T3, that is formed after the coupling of MIT and DIT, and also it will be containing MIT and DIT separately. Again, the same enzyme thyroid peroxidase is required for coupling reaction, that is coupling of MIT and DIT, resulting in the formation of T3, T4 and reverse T3. Next is proteolysis of thyroglobulin. So now you have seen thyroglobulin present within the colloid, that is present within the lumen. So the thyroglobulin contain MIT, DIT, T3, T4 and reverse T3, and they can store these thyroid hormones for around 2 to 3 months. Now by a process known as pinocytosis, the thyroglobulin will be taken into the cell. So pinocytosis is a type of endocytosis by which thyroglobulin present in the colloid is taken into the cell. So colloid droplet is reaching the cell. Now proteases present in the lysosome of the thyroid cell will cause digestion of the thyroglobulin. So thyroglobulin is a protein. So proteases are used for the thyroglobulin digestion. Now after digestion it will release the content, that is T3 and T4 and MIT and DIT. This T3 and T4 after releasing, it will reach the capillaries through the basolateral membrane. MIT and DIT will be released from the thyroglobulin, but it will not be secreted into the blood. It will remain within the thyroid follicular cell. Now the last step is secretion of thyroid hormone. So we know that thyroglobulin have reached the thyroid follicular cell by pinocytosis. Now the enzyme protease will cause digestion of the thyroglobulin, leading to the release of MIT, DIT and T3 and T4 that was present within the thyroglobulin. Now you can see in the diagram this thyroid hormones, that is T3 and T4, diffuses through the basolateral membrane of the follicular cell and they reach the surrounding capillaries. Then the MIT and DIT, they are released from the thyroglobulin, but they are not secreted into the blood. They will be present within the cell, and another enzyme, that is iodotyrosine deiodinase, will act on them and it will split them into iodine and tyrosine. Now both iodine and tyrosine are used for the thyroid hormone synthesis. That is, tyrosine is used for the synthesis of thyroglobulin and iodide is used for the synthesis of new thyroid hormones. And thyroglobulin is also not secreted into the blood. So when you get a question on thyroid hormone synthesis, you have to write under 6 headings, that is iodide trapping, that is taking iodide from circulation into the thyroid follicular cell, then conversion of iodide to iodine, that is iodide is converted to iodine with the help of enzyme peroxidase, then organification of thyroglobulin, that is attaching iodine to the tyrosine residue present in the thyroglobulin, again the same hormone, that is peroxidase, then coupling, that is attaching MIT and DIT, combining MIT and DIT resulting in the formation of T3 and T4, that is again mediated by the enzyme thyroid peroxidase. Then proteolysis of thyroglobulin, that is digestion of thyroglobulin by the enzyme protease, releasing the T3, T4 and MIT and DIT. Then finally secretion of thyroid hormones. By diffusion, thyroid hormones will leave the thyroid follicular cell and they will enter the capillaries. So you have to write the entire 6 steps explaining each step, and you can draw a neat label diagram showing every step, that is 1 to 6 steps of thyroid hormone synthesis. Hope it's clear. Thank you.